and one commentary. After that, we will have some time for common discussions before we start work in small groups. And in those small groups, the basic idea is to generate ideas for future international cooperation. And we will then we will have after these small group discussions, we will have some common discussions together and try to find out the key findings from each group. And this workshop ends no later than 4 p.m. And some basic guidelines for this workshop or for this meeting about sound. Please avoid extra noise via your microphone. Keep it muted if you are not talking. Videos, if possible, keep your cameras on. Especially when you are working in breakout rooms in those small groups. And we use chat for communication. And in case you drop out, use this invitation link again. And during those small groups works, we use breakout rooms and we utilize those rooms automatically. So you move in and move out automatically from those rooms. And before we start with opening words, we are going to do a short check-in exercise. And what does this mean is, so take your phone or your computer browser, go to menti.com and use the code 4842034. And there will be 2 different kind of questions available. Okay, and then Christian, it's your turn.
Thank you, Jari. Well, also from my side, a very warm welcome to everyone here who has made it. We had a lot of people signed up, and I don't really can see how many we now have, but it's a good crowd anyways. The main focus of this workshop, as Jari pointed out, is to interactively discuss international coordination and collaboration in the implementation of molecular monitoring methods. We will have a very interesting keynote speaker today from Professor Gernot Segelbacher. He will talk about new tools, new challenges, and how to improve knowledge transfer. Gernot is a professor of conservation genetics at the Wildlife Ecology Management at the University of Freiburg. He is also a chair of the IUCN Conservation Genetics Specialist Group and a member of the technical subgroup of the IUCN Task Force on Synthetic Biology and Biodiversity Conservation. After Gernot's talk, we will have a commentary from our very own Petteri Vihervaara, who is the leader of the Finnish Ecosystem Observatory project, and then we will have a short opportunity for a short discussion. And at 2:45, we will enter group discussions. But before we move on with the project, the program, I would rather that Veera Norros, who was leading the drafting of the national eDNA strategy, would say a few words about it.
Thank you, Christian. Can you all hear me and see the slide? Wonderful.
Yes.
So I'm Veera Norros from Syke, and I just wanted to take this opportunity to very briefly advertise our new roadmap for implementing environmental DNA and other molecular monitoring methods in Finland. So during the last year, we have been preparing this kind of strategy paper in which we summarize the state of the art of the implementation of these methods in Finland, and then identify some key development areas and suggest some concrete measures that should be taken in the next couple of years to proceed with the implementation. And our vision that we state in this paper is that molecular monitoring methods could be in extensive routine use in Finland by 2030. And to work towards this goal, we are kind of now hoping to enter this kind of coordinated transition phase in which we hope to get cross-governmental funding for testing molecular methods alongside traditional monitoring. Then we, of course, in international collaboration, would like to advance method standardization, promoting the service market, and then also, importantly, we're working towards establishing a national data management system. And I'm not saying any more about this roadmap now, but if you're interested, the first draft of this report is already online, and Tiina, I guess, has already posted the link in the chat, and if not, she will do it very soon. This draft is already fairly close to the final version, which will be published during this spring. So stay tuned and have a great workshop. And now welcome, Gernot.
Thank you, Christian. Thank you, Veera. And now, Gernot, it's your turn.
So now I don't see anyone anymore, but only my slide. I think you should see my slides and you should hear me.
Yes, we do.
Yes. Okay, great. So thanks everyone, and thanks Christian for joining, and thanks Christian for inviting me. It's always a pleasure getting at least virtually to Finland, being there for some undergraduate studies nearly 30 years ago. It's always nice to hear Finnish language, and I'm looking forward to the coming workshop and also to discuss things with you. So as you might see, I've changed the title slightly to the announced one. So it's now called New Tools and Old Challenges, because when I went through preparing the slides, I noticed that a lot of those challenges we've been through for quite some time, and I want to start with what we see regularly in the newspapers now. This is just kind of out from the press in early January report. You might have seen that that air eDNA sampling is getting more fancy. I know in Finland this has been done also with fungi and with plants. There's been publications last year as well. So techniques are getting more advanced and more fancy, and they got also a lot of traction in the public, and there's always media coverage on that because it's obviously interesting to sample air in a zoo and then get all kind of different animal DNA traces in there. So I've had a look on papers, and that's what's been published in the last a bit more than 10 years, and you see kind of a really increasing development. We're getting probably close to 1,000 papers on eDNA methods in this year, so it's going to be an exciting time. And I know you probably seen this picture already. This is kind of what we do with environmental DNA. Is kind of assessing species from different sources, maybe terrestrial, but mostly aquatic, could be also historic from soil or ice. So we'll have a kind of a wide variance on organisms we might eventually cover. And then obviously the technique is one side. So it's the applications we are interested in. And that could be kind of reconstruct or get some information what happened before. We are involved or getting, and that's probably the earliest studies on diet analysis, invasive species detection, and so on. Interesting in this graph from 2019, I found that monitoring or biodiversity assessment itself was not placed there. But we now have this guidance document available, which is actually also published early this year by Christian and others, which gives kind of a really clear outline how you should proceed. And I don't want to get into all the tech stuff today. That would be kind of another 3 hours probably. Just mentioning that as a really important resource out there. The question, however, is can we really use eDNA for monitoring biodiversity? We know that we can get traces, we can that, but how do we do that in the mid or long term? There's been a couple of fancy articles recently, also using nanopore sequencing technologies, saying, yeah, well, we can do that, and it gives a lot of traction from practitioners, and especially in countries in which are not that probably well equipped with laboratories and funds, South America, Asia, Africa, and obviously also leading to the discussion how much of these techniques should we use and allocate, and a lot of funding is going into these techniques as well. On the other side, we'll face a couple of challenges from the technical side. This is just kind of a short blog post from a few days ago, and this is kind of the IUCN issue brief on digital sequencing information, also posted in January. So whatever we generate on data, there's a whole lot of consequences we need to deal with, which are actually debated now in the ongoing CBD discussions as well. So this is also something we're not going to getting too much into detail today, but you need to keep that in mind. That is something which is happening. I want to go back from eDNA purely into how genetics has been actually getting into policy and management a couple of years back. And what you see is kind of major research milestones above the line, and you see potential policy milestones below the line. And in each of these milestones, I've been highlighting here genetic plays a crucial role. So we can kind of, obviously, genetics really got a lot of attention when PCR was invented, when DNA fingerprinting get into routine practice. So in the beginning of the 90s, so Conservation Genetics as a journal has been founded 2000, and already 2010 it was quite clear that genetic diversity itself is neglected in many international kind of negotiations and agreements. We have some genetic mentioning in the CBD targets in 2010. And at the same time, a couple of organizations have been developing. So it's the Conservation Genetics Specialist Group within IUCN. I'm chairing with Mike Bruford in 2014. And a couple of other organizations also kind of thought, okay, we need to do something about that. And many of you might be aware of that. When you go to a conservation or ecological conservances, genetics mostly is kind of on a side event. But this is about to change. So in recent years, that has become more and more relevant. And kind of what happened kind of the last year, that's actually from an organizational point of view, the so-called Coalition for Conservation has been founded. And it's actually linking the main initiatives from Society of Conservation Biology, from GEOBON, also from IUCN, to kind of discuss and interact and distribute also resources. And at the other side, obviously, there's been also a bit of scientific output in that. So when we go back to the early times, and many of you might know that, when we've been dealing with microsatellites, are not really large genomic data sets, it was always kind of a hassle if you would like to standardize across labs. And that's still difficult. Even if you agree on a same marker set on STRs, getting the similar results in a different lab often is really tricky. And probably human forensics were the first who had, obviously, they wanted a unique human database. They were probably the one who started very early and said, we need kind of a worldwide unique database and have to have that standardized. So this is just kind of a picture of kind of those loci on the chromosomes, human chromosomes they identified. And even in the current, I just found that on the web, the FBI quality standards, they refer to their standards on what you have to do in the labs to really get reproducible results. So standardization across labs has been difficult and is still an issue, I would like to say. Another lesson, I think, which is quite important is from large carnivores. And I've just took two pictures here. The one is the always ongoing discussion in the carnivore community. Is there dog-wolf integration, and what does that mean for policy and management? And you can see in that paper from last year a data set kind of pulled from Finland, Russia, also German and Romanian wolves, but also from the Iberian Peninsula, and then a lot of dogs. Obviously, if you want to pull these large data sets, you need to have a standardized methodology. Otherwise, you can't generate that. And that, especially for large carnivores, there has been several attempts to homogenize that, because we have the situation, for example, in southern Germany, we have a wolf that might have been shot, or you find some traces. Then you take an example of example form carcass, and then you send it to the laboratory in Frankfurt, which will confirm it is a wolf. And then they will relate it to the northern German. That's why I put in the map here, kind of the eastern northern German wolves here and say, okay, that might be that wolf coming from that and that pack. But if it's coming from the south, meaning from Switzerland, we don't have any possibility to do that in Germany at all. We have to send that sample to Switzerland, so they will do the parentage testing in their country. And obviously in large carnivores, which cross borders easily, that is really a tricky thing. I know that for bears in Norway, Sweden, Finland, and also for wolves, that has also been done. But it's been a very, very long way in standardizing that across countries, and it still doesn't work perfectly in many areas. So I think another key message, which we know we've been dealing with for quite a long time, is that we need a standardization across countries as well. Then we also have the issue that often biodiversity assessments are really taken on a national level. So that means we have our national monitoring system targeted on specific habitats or specific species. And then on the other side, we have the reporting system to the CBD. And interestingly, we've been published this paper last year on mostly focusing on genetic issues on CBD reports, but going through kind of all these 150 reports, we noticed that there's a large heterogeneity how individual countries report their data to CBD and as an obligation, and there is no standard whatsoever there. So a collaboration across borders, I think, is something we really urgently need on different levels. So one of my key messages would be we need more guidance and standards. While there is a lot of guidance out there on the technical side, we need also standards on the database and on the reference databases when we're talking about eDNA levels as well. One approach to do that, and this is just one, there could be others, would be the IE BioAtlas. Some of you might have heard of already, where kind of data are collected at a central place. And another one more on a national level would be developing specific pipelines. So in Switzerland, for example, they have had this approach. Okay, if you want to do an eDNA study of water, for example, for detecting amphibians, then there is a very strict protocol you have to follow. And you as a practitioner or kind of responsible for a specific area, you don't have to understand really everything what's happening in the lab, but you have kind of labs who do the stuff standardized, and that would be the gray area. And then you get back to the interaction with the practitioners on the others. Whoever is interested in, there's also a paper on how they do that. So developing pipelines for that, so that not every research project or kind of application has to go through the whole process again, seems to be quite important for me as well, if possible. And at the end of this talk, I also want to go a bit back to genetic monitoring versus monitoring of genetic diversity, which is actually two different things. I also did a quick search on are genetic tools used for biodiversity monitoring, just looking on genetics monitoring biodiversity. Again, an increase, which you would expect, but people often understand very different things. So what I want to talk about quickly is monitoring of genetic diversity. We all know we have ecosystems, we have species, we have genetic diversity as the three main pillars. But genetic diversity is not really monitored in many countries or been developed. There's only a few cases, because it means you measure genetic diversity for specific populations or species across time. So you have to have at least 2 time points. And we're doing currently a large meta-analysis on that. We've been producing a guidance document, which will be out in probably April or something like that. and that's not available. So this is just kind of showing. We'll highlight that. And I just want to make that point clear. Applying genetic tools does not necessarily mean you monitor genetic diversity. So while I think monitoring genetic diversity is one thing, using genetic tools to detect species or some other issues is something different. I think we have the potential to monitor genetic diversity as well. And we've been publishing also a paper on how we do that. So this is very, we're not going through all of that. This is very similar to the eDNA cookbook. So we have a number of lab-based steps. We have also non-genetic data, environmental data. Then we go through all the analyzing steps, and then we go to the databases. And then comes the big step, which means we want to have that into something meaningful. So we have to do a capacity building, and then it goes into policy and monitoring. And also that needs quite a bit on standardization in that synthesis as well. Last point here would be, if you develop a monitoring program, you definitely want to have the stakeholders' perspective, and there's been an attempt and a paper in Switzerland as well. So these are the factors which are the ones which are seen as most crucial if you're developing a monitoring program. And you see, obviously, there is data centers, central data facilities. You have research institutions, obviously. But then you have also the federal and regional authorities. You need to have a large scientific network, and so on. So whoever is interested can dig into that as well. So the question is, do we want to have a bottom-up or a top-down approach in doing that? And I think if we sit together and plan this kind of approaches, the success, and that's my experience, and also kind of talking with those people who have been working in the eDNA community from the very beginning, it means you need to start building up partnerships and trust from the very, very beginning with many different partners. So it's not all about the techniques, which themselves are still having some issues, but it's also very much relying on kind of this interaction part. So let's sum up. So eDNA is a methodology in development. Let's don't forget that. It's improving all the time, and we'll see where we will be in 5 years from now. The second point is assessment with DNA needs standardization, not only in the methods, but also quite crucial on databases and in reference libraries, so that can be compared across areas and countries. And the third point might be monitoring needs really involvement from many parties from the beginning on and across borders. And with that, I wanted to give you a short introduction where this way might be, and I hope I got you some food for thoughts, and I'm looking forward to the discussion.
Great stuff, Gernot. Great stuff. Really, I think you touched on very important stuff like trust and the need for more standards in order to build that trust. But now let's hear from Petteri and his commentary.
Petteri, now it's your turn.
Okay, thank you. Can you hear me?
Yes, we can.
Good. Okay, thanks, Gernot. It was very interesting presentation and very important topics that you were telling to us. So it's my pleasure. I just heard half an hour ago that I have to give a commentary speech to your presentation. So I was quite enthusiastic to see your presentation, but there were many points which are reflecting very well what I'm working with. So basically, if I start from this global collaboration, so you mentioned GeoBond and CPD negotiations, and I think it's very relevant framework for all the discussions what is happening then on the regional and national scales, as well as across the scientific networks. So I think that's really important point that you highlighted, that at the moment, for instance, these national biodiversity monitoring schemes and NB subs, they are not really focusing for genetic monitoring, but it should be having more and more importance at the moment when there are these CBD negotiations ongoing, that these are really tools that should be included and developed during the coming years. And also I think it's one relevant point of these CBD negotiations that despite of getting overall global biodiversity framework for different biodiversity goals and targets, it's very important that at the same time we are paying a lot of attention that we also monitor what's happening in the nature itself and the biodiversity, and not only these high policy level targets and how the process is developing, but at the same time we need to improve our capacities to monitor that how these policy impacts are affecting to the biodiversity itself. And that needs a lot of collaboration. I think this kind of eDNA and molecular biological methods could really play important role in that sense. Then coming back to the European scale, so maybe you are aware that GeoBon is playing also a role in the different regional and national biodiversity monitoring schemes and their development. So currently there's EuropaBon project that is very significant project for European biodiversity monitoring community. And parallel to that, there's also a new European biodiversity partnership, Biodiversa Plus, which is working in close collaboration with this EuropaBon project and European bodies. And maybe those are the two key initiatives at the moment which are also having possibilities to influence this kind of implementation of new methods and standards, how they should be taken into consideration in the future. And actually, I would like to maybe continue discussion with you and other colleagues later with the smaller groups that what would be the expectations towards this EuropaBon and Biodiversa development in the future, because they have at the moment there are good possibilities as well, some resources to pilot and test how to get these best practices and standardization work in a different countries and different ecosystem types as well. But yeah, I just recommend you to check out EuropaBon and Biodiversa Plus if you are not aware of those yet, and maybe we can continue that discussion later. And then finally, maybe coming back to the really national scale, colleagues Aapo and Veera and others are at the moment in Finland developing national biodiversity strategy and also thinking about how to build up new indicators on a national scale. So that's one thing, that how we could emphasize the need for this genetic diversity monitoring also across this kind of national indicator systems. And that's it would be really interesting to see your guidance document, which you mentioned that is coming April 22 out. So maybe you can inform us when it's published and available. And anyway, I would like to encourage those who are working with the national biodiversity strategies and indicators to also be open for starting from a blank table to also include this kind of new currently missing indicators for national monitoring schemes. So I think that's really interesting possibility as well. So that's all so far. Maybe one last thing, as I'm not really in-depth expert of any kind of DNA methodologies, but thinking in a general level, these different monitoring schemes, I think it might be maybe useful sometimes to think that how these different facets, like if you are taking bulk samples from air or water or terrestrials, how these could be then applied across different habitats, even around different regions of the planet, that if there are this kind of methodological advantages, it would be really useful to maybe look not only your own expertise, habitat or facet or biodiversity group, but also to think that how apply these methods across the different schemes, it would be interesting as well. But thanks for a good talk and looking forward discussions later in smaller groups.
Thank you, Petteri. Now we have some time for common discussions. So if you have any questions, any comments, please use that your raise hand function so I can give you your turn. Seppo.
Yes, thank you everybody. Greetings from Oulu Quantum Institute, where we also address among physics and space, I mean other multidisciplinary questions, the kind of ecosystem and the species diversity issues. So I was just I missed a little bit at the beginning. Maybe there was discussion about these things, the databases, and like this eyeball program that is also kind of offering these DNA tags for species identification based on the mitochondrial sequences, and then how these are related, let's say, together concerning what we just heard from Gernot's talks, and then concerning what Petteri was outlining, so that there are, I mean, I guess the species, but we have lots of species within species. So then concerning about the kind of microbiomes and variety of sort of hosts carrying distinct DNAs and their classifications, and how this might create issues on this process altogether. And then what we are targeting basically is a kind of a natural big one, nano and micro level communication aspects that are then also using these nucleic acids as a way to transmit information, and that's something that I'm wondering how this should be considered in terms of these programs. But I mean, collectively, these resources and the reference databases, obviously, and the genetics on those is very essential. And I mean, concerning about the European process, but also more broadly. And I don't know, I'm not an expert here, but just asking question how this is somewhat developing. Maybe Petteri or Gernot or others could maybe comment on this. Thanks.
Okay, Gernot or Petteri, would you like to comment?
I think this is a tricky question, and I think the reference databases is really an used issue, because we sequence and then we get a lot of sequences, and not all sequences will match to species, and that might be higher levels, but it might be whatever we call MOTUs, operational taxonomic units. So for example, red list IUCN species is getting already quite complicated. So what are we going to conserve here? So that really is an issue, and obviously all reference databases only work if we have a reference in the database already. So if we sequence something and then we find something new and we don't have a reference yet, then so we have no information. So and if we just screen, and I think I like the idea Petteri pointed out, so you're just going into another area and then kind of thinking outside the box and saying, okay, how can I apply that? In some areas you have to establish your reference database first. Just a quick example. So we've been working on wood. So the idea was getting beetles in wood. Obviously there's a large Coleoptera database available, but whatever we got from sequencing was everything else than beetles, all kind of noise in there. So it's not always quite meaningful what you get out there. So my plea is we need, for each study, we need kind of a reasonable database, and they obviously should be also harmonized. And I think one thing BioAtlas tries to do that somehow to set up these databases. But then obviously there is many other databases. BOLD, you have the GeneBank, you have whatever databases, and I think there's more homogenization necessary here.
Okay, and then Aapo. You have some
Thank
comments
you.
or questions?
Yes, I have some. Thank you. I think it was very nice to see that you highlighted the fact that when you use molecular genetic tools, it doesn't necessarily mean that you're actually measuring genetic diversity. And this is something that's very important to highlight when the adoption of the different molecular methods becomes more commonplace, especially in practice, in practical conservation. But that also raises some thoughts that how I'm wondering if large-scale genetic diversity measurements across species, across multiple groups of different species, how feasible that is, and if you consider that we have many countries that have different levels of development when it comes to genetic methodology, and then added on top of that the fact that genetic diversity conservation goals might actually conflict those at the species diversity level. So I'm wondering if there are any, you must have thought about this, but what are the indirect ways of measuring genetic diversity, and have these been applied what is the kind of current status of thinking about indirect genetic diversity indicators apart from census population sizes?
Okay, so I'll break that down to the two questions. Obviously, at the moment we do have 3 examples where genetic monitoring programs have been established in sensu stricto, so it means we are monitoring genetic diversity, and this is Switzerland, Sweden, and Scotland, with 2 approaches kind of on their way somehow in South Africa. And obviously, as you pointed out, that really depends how much resources and funds you are. In Switzerland, they have really agreed on 30 different species. They have sampled them across the whole country, and they will spend quite some amount of Swiss francs on that, doing that on a 5-year interval. When we've been producing this guidance document, we've been always hearing the comment, well, that's a good approach, but we can't do that. Like, is the response you get normally from South America or from Asia. So there are ways in getting, as you rightly pointed out, in deriving genetic diversity measures indirectly. Probably what we try to get into the CBD current negotiations, probably the most easiest way is the effective population size. Although it is highly debated how meaningful that might be, but that's something. And the number of unique populations, which is also a kind of a proxy, which is currently, we've been commenting on the 0 draft, well, and now it's kind of the 1 point whatever draft on CBD negotiations. So it looks like as we get some indicators and targets in there, which also might mean we can kind of indirectly do that, but it's not an easy one. But it's important to have that opportunity, even for those countries who can't spend millions on kind of sequencing whole genomes across all taxa, for example. But I would kind of, I'm happy to send you your copy. We are in the layouting process, but otherwise in April we should be done. So I think we break that down to these different points, exactly answering what you've been asking, Apple.
Okay, thank you. And the last comment from Kati.
Yeah, thank you for very interesting presentation. I just wanted to ask still these barcoding monitoring systems, are they more used beside the traditional monitoring of species in the field? And I was thinking that where there will be a turning point when these methods become more common or dominating in the field, observing species richness or species diversity? And can you see, is there a potential risk also if we are directing too fast for this, if this become very cheap also to monitor, especially in more developed countries, that we kind of totally ignore the more traditional monitoring of species in the field and jumping into these fancy techniques?
Okay, I give you my personal opinion on that, and others here might have other opinions. I think molecular methods will mostly be complementary to the current approaches. They will not be completely able to replace that. Although for some instances we have cool data which show okay, you get much more species, and that's why in many grants I've been seeing by reviewing and also by lots of discussions at conferences, people say, oh yeah, well, let's all barcode everything and then we have the unique solution. I don't think this is going to work that way. And we also know that with genetic methods, we will overlook some traditional kind of monitoring results. So I think we need to think of a complementary approach, and that's why I would really be cautious in saying, okay, let's replace everything. I think from the matter of finances, obviously there's a large tendency also with using this handheld portable devices and let people go out there using and doing the sequencing out in the field, so you don't have all the issues also in more remote islands or areas. But I honestly don't think, but that's probably also because I'm kind of more like a field person originally. I don't think it's going to be completely replaced. And I always would warn to trust completely. Okay, there is several studies, but I'm looking at the time. We're not going to explode it, but I think we know from, especially from amphibians and crested newt examples, that the traditional kind of monitoring scheme often is still having quite it's still quite valid.
Okay, thank you. I stop now this discussion. We have to move to small groups, and you can continue your sharing your ideas and other comments there. And after that, we also have this common discussion part, so you can hold your questions in your mind, and we can continue then. But what we are going to do next is so that we are going to work in small groups. We are going to utilize